So in this video, we are going to talk about the structure of DNA, which was demonstrated by Watson and Crick. And this model is nothing but double helix model. And the first point is, according to this double helix model by Watson and Crick, the DNA is double-stranded. DNA is double-stranded. That means DS. There will be two strands. Two strands of what? Two strands of nucleotides. Two strands of nucleotides. Two strands of nucleotides. And the second thing is, these two strands will be anti-parallel to each other. Like, for example, if we are going to step into the staircase. You observe there are two railings, right side and left side. There will be two railings and steps. In the same way, these two strands of DNA also runs parallel to each other, but they never cross each other. These two strands of DNA will run parallel to each other. They never cross. As if you see the two strands. So this is one strand, and the other strand runs parallel. But they never meet each other, they never cross each other. Okay, and this anti-parallel polarity means, suppose one strand 5' to 3', the other strand 3' to 5'. So I'll tell you, it's very simple. What is this 5' and 3'? Suppose you take this strand, the blue color strand which I have drawn, the 5' means, take the nucleotide in this phosphate group. This 5 carbon, 5th carbon, so 1, 2, 3, and 4, and this is 5. The 5th carbon will be free. And the next, let's say this nucleotide. Let's take this nucleotide now. First carbon, second carbon, third carbon. This third carbon will be free. Here, generally, OH functional group will be present. This is first carbon, second carbon, third carbon, fourth carbon, and 5th carbon phosphate group. And this phosphate group will be attached to this functional OH group of the above nucleotide, right? That bond is nothing but phosphodiester bond. This bond is called phosphodiester bond. So here, 5', so 5th carbon is free here, and here third carbon is free, so 5' and 3'. In the same way, the other strand, why it is showing 3' and 5'? It's very simple. Here, now it has to be inverted. Now, which is the first carbon? This could be the first carbon, and next number 2, number 3, number 4, and number 5. So here 5th carbon is 3, so 5'. And coming to the before that. So here first carbon, second, third, fourth, and 5th. And here already there is OH group, and here phosphate group. Both are attached by phosphodiester bonds. And here the third carbon is free. So the two strands will be possessing anti-parallel polarity. So 5' to 3', 3' to 5'. So this is called anti-parallel property. And coming to, we already know that nitrogenous bases attached to the first carbons, right? So here the first carbon, nitrogen, and here also first carbon, nitrogen. So these nitrogenous bases are, adenine will be attached to thymine by two hydrogen bonds. The adenine will be attached to the thymine by two hydrogen bonds, and cytosine will be attached to guanine by 3 hydrogen bonds. Let's say this nitrogenous base is adenine. Let's say this nitrogenous base is thymine. And there are how many hydrogen bonds will be? 2 hydrogen bonds. And let's say this one, here the nitrogenous bases are cytosine and guanine. Okay, 3 hydrogen bonds. As I said in my previous video, there are 3 types of pyrimidines. That uracil is not present in the DNA, and that uracil is replacing the thymine in RNA. In the RNA, there will be uracil, which is going to replace this thymine. So thymine is unique for DNA, uracil is unique for RNA. And coming to the next point, if you draw an imaginary line, here, we can understand that there is particular diameter for this DNA helix. There is a diameter. Let's say this is one helix, and the diameter of helix is 20 angstroms. The diameter is 20 angstroms. Diameter is 20 angstrom. And these grooves are nothing but, these 2 grooves are nothing but shallow grooves. And these are nothing but deep grooves. These are shallow and these are deep grooves. The distance between two shallow grooves, is 34 angstroms. 34 angstroms. This is nothing but one turn. 34 angstroms. And each turn, one turn is nothing but the region of DNA strands, which is covering from one shallow groove to the other shallow groove. This is nothing but one turn. And each turn of DNA consisting of 10 base pairs. 10 base pairs. See, this is one base pair and this is another base pair. Like that, 1, 2, 3, and 4, 5, 6, 7, 8, 9, 10. So like that, 10 base pairs will be present. Here they are coiled. Here we simplified on one beside the other. I have drawn here to understand how the nucleotides are arranged to each other in 2 strands. This nucleotide sequence is of one strand and this nucleotide sequence is of another strand. So in this way, the structure of DNA was explained by Watson and Crick, and this is called double helical model, which was proposed by Watson and Crick. Again, I'm going to repeat the points. First thing, according to double helical model by Watson and Crick, the DNA is a double-stranded DS. DS stands for double-stranded. And next thing, they're consisting of 2 strands of nucleotides, 2 strands of polynucleotides, and they're consisting of nitrogenous bases, adenine, thymine, cytosine, guanine, Adenine thymine double bond hydrogen bonds, cytosine guanine triple bond hydrogen bonds. And coming to this two strands, anti-parallel polarity. These two strands are anti-parallel polarity. Why? Because one strand 5 prime to 3 prime, the other strand 3 prime to 5 prime. Why it is 5 prime? Because here fifth carbon is free and the 3 prime is third carbon is free. See here OH group is free and here phosphate group is free. And coming to the other strand, the parallel strand which is running parallelly, so this one. Let's take 3 prime here I will take. Because here third carbon is free, that means OH group, right? OH group will be free. And here fifth carbon is free, that means phosphate group. Phosphate group is present at fifth carbon, that is free. So this is anti-parallel polarity. Coming to nitrogenous bases, we already discussed. And if you draw an imaginary line at the center of this DNA strands, the diameter will be 20 angstroms. The diameter will be 20 angstroms. And apart from that, there are shallow grooves are there, deeper grooves are there. The shallow groove to one shallow groove is nothing but 34 angstroms, 34 angstroms. And between one base pair to the next base pair, it is one more point added here, 3.4 angstroms. The one base pair to the other base pair is 3.4 angstroms. So this is all about the structure of DNA. Hope you all understood. So please like my video